Good day, my student. Today our lecture about doctrine of infection. Doctrine about infection studies the properties of microorganisms that provide their viability in the macroorganism and their ability to produce a pathogenic effect on it. It also studies the defense and adaptation reaction of macroorganisms to this effect. In fact, the presence of some microorganisms can even benefit the host. Therefore, before we discuss the role of microorganisms in causing disease, let's examine the relationship of microorganisms to the healthy human body. Relationships between the normal microbiota and host cells neutralism, when the patterns do not produce any effect on one another. The relationship between the normal microbiota and the host is called symbiosis, a relationship between two organisms in which at least one organism is dependent on the other, and includes different types of relationships. Commensalism, one of the organisms benefits and the other is unaffected. Example of this on this relationship, for example, butterfly and flowers. Mutualism is a type of symbiosis that benefits both organisms. Example of mutualism is shark and small fish. And interesting for us, parasitism. Parasitism, relationship between two species in which one benefits at the expense of the other, sometimes without killing it. But interesting that relationship can change under certain conditions. Classification of form parasitism. Microorganisms can be obligate parasite, are enabled to exist outside the host's body, as they had lost all their ability in nutrition and can multiply only in the intracellular kind of existence. Example of obligate parasite means bacteria, rickettsia, chlamydia, and viruses. It's obligate parasite. Can't multiply, can't grow without host cell. Obligate parasite can be intracellular, penetrate into a cell and multiply inside of host cell, and extracellular parasite attached to cell surface and spread along intracellular areas. Next group of parasitism, opportunistic pathogens or facultative parasites, can only cause disease in situations that compromise the host defenses, such as the body's protective barriers, immune systems, or function normal microbiota, and are able to exist for some time in an environment outside the host body. Opportunistic pathogens or opportunistic parasites can be intracellular parasites, can live and multiply both inside a cell and in an intracellular area. For example, pathogens of tuberculosis, causative agent gonorrhea, or meningococci infection. And extracellular parasites can live and multiply in environment. And last, saprophytes is not interesting for us because are microorganisms which utilize dead organic substrate. On getting into a human body, some of them may lead a parasitic mode of life, became pseudoparasites, but this phenomenon is sporadic. What does it mean? Definition: infection, infective process, or infectional disease. Infection is the totality of the process arising in interaction between the microorganism and macroorganism. Infection is the invasion or colonization of the body by pathogenic microorganism. Infective process or infections process is the totality of pathological, physiological, respiratory and other responses of the microorganism. Infection disease is a totality of clinical and laboratory symptoms that arise as a result of response to the intrusion of a microorganism. Disease occurs when an infection results in any change from a state of health. What does it mean? Symptoms or change in body function such as pain and malaise. They subjective changes aren't apparent to an observer. The patient can also exhibit signs, which are objective changes. The physical can observe and measure. Frequently evaluated sign includes lesion, swelling, fever, and paralysis. Specific groups of symptoms or signs may always accompany a particular disease. Such as group is called syndrome. Koch's postulate they are summarized as follows. First, the same pathogens may be present in every case of the disease. Second, the pathogen must be isolated from the disease host and grown in pure culture. The pathogens from the pure culture must cause the disease when it's inoculated into a healthy susceptibility laboratory animals. And pathogens must be isolated from the inoculated animal and must be shown to be the original organism. You can see postulate Koch's on these pictures. Next, the prevalence of the disease is the number of people in population who develop a disease at the specified time, regardless of when in the first appeared. If particular disease occurs only occasionally, it's called sporadic disease. Example, typhoid fever. A disease constantly present in a population is called an endemic disease. For example, common cold. If many people in a given area acquire a certain disease in the relatively short period, it's called an epidemic disease, influenza. An epidemic disease that occurs worldwide is called pandemic disease, COVID-19. Another useful way of defining in scope of the disease in terms of its severity or duration. An acute disease is one that develops rapidly but lasts only a short time. A good example is influenza. A chronic disease develops more slowly. The body's reaction may be less severe, but the disease is likely to continue or recur for long periods. Example, infectional mononucleosis, tuberculosis, hepatitis B fall into this category. A subacute disease, a disease that is intermediate between acute and chronic disease. And this disease characterized by disseminated intellectual function and loss of neural function. And latent disease is the one in which the causative agent remains inactive for a time, but then became active to produce symptoms of the disease. An example is shingles, or of the disease caused by varicella virus. Infection can also be classified according to extent to which the host body is affected. Local infection is one in which the invading microorganisms are limited to relatively small area of the body. Some examples of local infection are boils and abscesses. In a systemic generalized infection, microorganisms or their product are spread throughout the body by the blood or lymph. Example, measles viruses can cause systemic infection. Very often, agents of local infection enter a blood or lymphatic vessels and spread to other specific parts of the body where they are confined to specific areas of the body. This condition is called a focal infection. Focal infection can arise from infection in areas such as the teeth, tonsils, or sinusitis. Sepsis is a toxic inflammatory condition arising from the spread of microbes, especially bacteria or their toxin from focus of infection. Septicemia, also called blood poisoning, is a systemic infection arising from the multiplication of pathogens in the blood. Septicemia is a common example of sepsis. If bacteria present into blood, it's conditionally bacteremia. Toxinemia refers to the presence of toxin in the blood, as occurs in tetanus disease. And viremia is conditionally if viruses present in blood of your patient. A definite sequence of events usually occurs during infection and disease. For an infectional disease to occur, there must be reservoir of infection as a source of pathogens. Next, the pathogens must be transmitted to a susceptibility host by direct contact, by indirect contact, or by vectors. Transmission is followed by invasion, in which the microorganism enters the host and multiply. Following invasion, the microorganism injures the host through a process called pathogenesis. Development of disease or stage of infectional disease. Incubation period is the interval between the initial infection and the first appearance of any signs or symptoms. Prodromal period is a relatively short period that follows the period of incubation in some disease. The prodromal period is characterized by early or mild symptoms of disease, such as general aches and malaise. Period of illness. During the period of illness, the disease is most severe. The person exhibits overt signs and symptoms of disease, such as fever, chills, muscle pain, myalgia, sensitivity to light, photophobia, sore throat, pharyngitis. If the disease is not susceptibility overcome, the patient dies during this period. Period of decline. During this period of decline, the signs and symptoms subside. The fever decreases and their feeling of malaise diminished. During the phase, which may take from less than 22 hours to several days, the patient is vulnerable to secondary infection. And last period of infectional disease, it's period of convalescence. During the period of convalescence, the patient regains strength and the body returns to its pre-disease state. How infections spread between human population? Human reservoir, these people with sign and symptoms not a disease may transmit the disease. In addition, some people can harbor pathogens and transmit them to others without exhibiting any signs of illness. There are people called carriers, are important living reservoir of infection anthropognosis. Animal reservoir, both wild and domestic animals are living reservoirs of microorganisms that can cause human disease. Disease that occur primarily in wild and domestic animals and can be transmitted to humans are called zoonosis or zooanthropognosis. And non-living reservoirs, the two major non-living reservoirs of infection disease are soil and water, and these disease groups of disease sapronosis disease. Route of transmission, infectional disease. Contact transmission is the spread of disease agent by direct contact, indirect contact, or droplet transmission. Direct contact transmission, also known as person-to-person transmission, is a direct transmission of an agent by physical contact between source and susceptible host. Indirect contact transmission occurs when the agent of disease is transmitted from its reservoirs to a susceptible host by means of non-living object. Droplet transmission is a third type of contact transmission in which microbes are spread in droplet nuclei that travel only a short distance. Fomate vesicle transmission is the transmission of disease agent by medium such as water, food, or air. Waterborne transmission, foodborne transmission, and airborne transmission. Vectors. Arthropods, ticks, or mosquitoes are the most important group of disease vectors. Animals that carry pathogen from one host to another. Intrauterine or placental route of transmission from mother to fetus through placenta, and vertical route of transmission when the fetus passed through the infected birth canal of the mother. On this slide you can see type of transmission, route of transmission, direct contact. Preventing direct contact transmission through the gloves, masks, and face shields. Infected contact transmission, droplet transmission, vectors transmission from animals to human body. We know vectors, for example, ticks or mosquitoes, and through water, food, air, and intraplacental route of transmission from mother to fetus. On this slide you can see how infection disease can transmit: person to person, droplet, fecal-oral, airborne, fomates, and insect bite. The role of microorganisms in the infection process. Pathogenicity is the potential ability of microbes to cause an infectional process. This is a specific genetically determined trait. According to the degrees of pathogenicity, all microbes are divided into three groups: non-pathogenic or saprophytes, conditionally pathogenic or opportunistic microorganism, or potentially pathogenic and pathogenic microbes. Virulence is the individual property of the given strains of microbe to cause the development on the infectional process. This is a measure of pathogenicity. Its quantitative and qualitative characteristics of phenotypic manifestation of the genotype of the pathogenic microbes. And the microorganisms classified by virulence like highly virulent, moderately virulent, slightly virulent, and avirulent microorganism. Avirulent microorganism we can use for creative vaccine. Determination of virulence. For determination of virulence properties of microbial cell, we use laboratory animals, susceptibility animals, and we study dose lethal minima. Is the minimal lethal dose for microorganism or their toxin which may cause death of the test animal within a definite time period. It is a relative value depending on an animal species. Dose lethal 15% is an amount of the microorganism or toxins causing death of 15% of the infected test animals. Dose certainly lethal is an absolute lethal dose in amount of microorganisms or toxins causing death all infected test animals. And last characteristic of virulence microbial cells, infectional dose ID, is the minimal amount of the pathogenic microorganisms that cause infectional disease in defined number of test animals. Infection doses don't kill animals, don't cause death of animal, just cause infectional process. What is it? Bacterial virulence factors. Many factors determine bacterial virulence or the ability to cause infection and disease. These factors help the microorganism to invade the host, evade host defenses, and cause disease. The following are type of virulence factors: adhesion factors, invasion factors, antiphagocytic factors, toxin production, extracellular enzymes, bacterial biofilms, and intracellular survival. Most groups of virulence factors. First, factors of adhesion and colonization. Specialized structures like pili that help their adherence to the host cell mucous membrane. Those allow terms to start the disease process. If they did not adhere, they will be steps away. Example, Escherichia coli have a pili to mediate the attachment to a urinary tract epithelium. The lipolytic acid causes adherence of the streptococci to buccal epithelial cells. Next group, factors of invasion or invasiveness factors. It's the ability of bacteria to invade the tissue and multiply. Invasion of the host epithelium is central to the infectional process. Next group, virulence factors, factors of aggression or aggressiveness or antiphagocytic factors. Some pathogens evade phagocytosis or leukocyte microbial mechanism by adsorbing normal host components to their surface. Next group, factors of toxigenicity or toxin formation, denote ability to generate exo and exotoxins. Factors of adhesion and colonization, example, fimbria, first order, proteins of the outer membrane, adhesive proteins, lipopolysaccharide, lipoteichoic acid of the cell wall, and other structure which may be on the surface of microbe or be part of capsule and cell wall. Factors of chemotaxis and mobility. Factors of adhesion help microorganisms attach specific cells, susceptibility host cells. Factors of invasion or spreading factors is a term for designation for a family of bacterial enzymes which disrupt the host cell of tissue and intracellular space. Exoenzymes of microorganisms. Example: hyaluronidase, neuraminidase, fibrinolysin, DNase or nuclease, collagenase, lecithinase, protease, and hydrolysates. It's enzymes which produce microorganisms, and these enzymes cause different effect upon cells of microorganisms. For example, Bacterium attach surface cells, invasive bacteria reach epithelial surface. Hyaluronidase destroy hyaluronic acid, its component of cells, and destroy of collagenase, and bacteria invade deeper tissues. Next, factors of aggression are unchanged resistance of microorganism to defensibility of the microorganism. Factors aggression can have function antiphagocytic function. Main antiphagocytic function is capsule of microorganism. Antiphagocytic factors includes enzymes like plasma coagulase, catalase and superoxidismutase, aminopeptidase, protease, protein A, and other factors, peptidoglycan, teichoic acid, and other components of the cell wall. For example, on this slide you can see bacteria produce coagulase. Coagulase forming clot. And bacteriolate produce kinase dissolving clot and releasing bacteria. If bacteria release, bacteria can ability to spread in organism of human. Next group, factors of toxigenicity or toxin formation. Toxins are either metabolic products of a microbial cells. If we know about exotoxins, produce gram-positive and gram-negative bacteria or integral components of a cell wall. Gram-negative bacteria that are released as result of its destruction, endotoxin, that bring in disorder into the life of the microorganism. Next, main features of exotoxin and endotoxin. You can see source: exotoxin, gram-positive and gram-negative bacteria. Source of endotoxin: outer cell membrane of most gram-negative bacteria. Secreted from cells present for exotoxin and secreted from cells known for endotoxins. Chemistry: polypeptide. Endotoxins is lipid A, components of lipopolysaccharide, and release after lysis of bacterial cells. Location: exotoxin located in plasmid or bacteriophage, endotoxin in bacterial chromosome. And I will not read this slide. We will study this slide in laboratory class, and it's for your knowledge. You have to know this characteristic, different features between endo and exotoxin. Exotoxin are secretory protein substances with fermentative activity which are produced by microbial cells. Exotoxin, I repeat, may produce like gram-negative and gram-positive microorganisms. Exotoxins are thermostable. They are highly specific in action. They are responsible for the clinical manifestation of infection process. Can act at distance, far from the focus of infection. Their effect is highly toxic. They exhibit high immunogenicity. In response to their injection, specific antibodies originate to neutralize to their effect. When treated with formalin, exotoxins get detoxified and are transformed into antitoxins or toxoid, and which are devoid of toxin properties but maintains an ability to induce antitoxic antibodies. I repeat, source exotoxin, mostly gram-positive microorganism and can produce gram-negative microorganism. Relation to microbes by product of growing cell. Chemistry proteins. Neutralization by antitoxin, yes. Fever can cause, cannot. And lethal doses, small. Classification of exotoxin by molecular arrangement. Complex, which are composed of two fragments, A and B subunit. B fragment interact with the receptor of sensible cell. Adherence is on surface and form transmembranous canal through which A fragment, the toxin proper, penetrates into sensible cell and exhibits its toxin properties. And plain or simple exotoxins are increased exotoxins. They synthesize in bacterial cells as protoxins and when are cut into A and B fragment by protease, they get transformed into active forms. On these pictures you can see mechanism and arrangement of exotoxin. Bacteria produce and release exotoxin, A subunit, a B subunit or B fragment bonding component of exotoxin bind to the host cell's receptors and exotoxin enters into the cells. A subunit, you see. A active components of exotoxin alters cell function by inhibition protein synthesis. Example, B components is released from the cells. By an extent of bonding with bacterial cells, we know A group are those which are secreted into an external environment. B group are partly secreted into the environment and partly bound with the bacterial cells, and C group those that are bound with the bacterial cells and released only after its death. Classification of exotoxin by the nature of targets. Neurotoxin, which damage nerve cells. Hemolysins, which destroy erythrocytes. Enterotoxins, which affect the cells of the gastrointestinal tract epithelium cells. Dermatotoxins affecting the skin cells. Leukocidin affecting leukocytes, neutrophils, and phagocytes. On these pictures you can see hemolysins, different type of hemolysins toxin, beta hemolysins, alpha and gamma hemolysins, and different type of hemolysins cause different effect upon erythrocytes. B hemolysins cause full destruction of erythrocytes. Alpha hemolysins destroy not full erythrocytes, and gamma hemolysins do not destroy of erythrocytes. Bacteria with exotoxin. Carinobacteria diphtheria or diphtheria toxin mechanism inactivate elongation factors Pseudomonas aeruginosa exotoxin A. Mechanism of this toxin inactivated elongation factors. Shigella subspecies produce Shiga toxin and enterohemorrhagic Escherichia coli Shiga-like toxin. Mechanism of this toxin inactivated 16S ribosome by removing adenine from rRNA. Increase fluid secretion. First group of exotoxin inhibition protein. Diphtheria toxin, Shiga toxin, and Shiga-like toxin. Exotoxin which increase fluid secretion produce enterotoxigenic Escherichia coli, heat label and heat stable toxin. This classified by sensitivity to temperature. Heat label toxin overreactives adenylate cyclase, increase cycle adenylate cyclase, increase chlorine secretion in the gut and water efflux. Heat stable toxin overactivates guanylate cyclase, increase cycle GMP, and decrease resorption of chloride natrium and water in gut. Bacillus anthracis produce edema factors. Mechanism action mimicries adenylate cyclase, increase cycle concentration cycle AMP. Vibrio cholera produce cholera toxin. Mechanism action overactivate adenylate cyclase, increase cycle AMP by permanently activating GS and increase chloride secretion in gut and water efflux. On this slide you can see two groups of exotoxin which cause inhibition protein synthesis but different mechanism of this process, and groups which cause increased fluid secretion with different mechanism. Next group, toxin which cause inhibit phagocytic ability. For example, pertussis toxin overactivate adenylate cyclase, increase cycle AMP by disabling G1 cells, impairing phagocytosis to permanent surveillance of microbe. Next group of exotoxin inhibit release of neurotransmitter. For example, Clostridium tetani produce tetanospasmin. Both are proteins that cleave snare soluble and as neurosystems attach proteins receptors, a set of proteins required for neurotransmitter release via vesicular fusion. And Clostridium botulinum produce botulinum toxin. But this toxin inhibit groups of inhibit release neurotransmitter have different mechanism of action. For example, tetanospasmin toxin prevalent release of inhibitory GABA and glycine neurotransmitter from Renshaw cells in spinal cord and cause clinical symptoms or signs like spastic paralysis, rhesus sardonic, and trismus, lockjaw. Botulinum toxin. This inhibit release of transmitted too, but mechanism actions of this toxin release of stimulatory acetylcholine, signals of neuromuscular junction and cause flaccid paralysis, HOPB symptoms. Bacteria contain exotoxin which cause lyse cell membranes. For example, alpha toxin, streptolizin O, which produce Clostridium perfringens streptococci pyogenes. Mechanism of alpha toxin phospholipase lecithinase that degrades tissue and cell membrane. Streptolizin O mechanism action protein that degrades cell membrane. And different groups of exotoxin super antigen causing shock. Staphylococci aureus produce toxic shock syndrome toxin, and streptococci pyogenes produce erythrogenic exotoxin A. And the mechanism action of this toxin super antigen causing shock cross-link beta region of TCR to MHC class second on outside of the antigen binding site and overwhelming release interleukin groups 1, 2, interferons gamma, tumor necrotic factors alpha, and cause shock of your patient. What does it mean endotoxin? I repeat, endotoxin cause just gram-negative microorganism. This structure of outer membrane, chemical it's lipid. Its structure lipid A of lipopolysaccharide of gram-negative microorganism, and endotoxin release after destruction of outer cell membrane of gram-negative microorganism. Endotoxin don't have specific activity upon cells like exotoxin and cause common symptoms. Example, pyrogenic effect, elevation of body temperature, because endotoxin induce release of interleukin-1 from macrophage, which affects the center of thermoregulation. Toxic effect on the vessels, as endotoxin enhance permeability of vascular walls. This results in hypotonic effect, in several cases a collaptoid state. Stimulating effect on the blood coagulation systems, as it activates Hemans factors. This condition is associated with microthrombs and impaired microcirculations. In several cases, syndromes of disseminated intravascular coagulation or DIC syndrome. Endotoxin cause cardio and hepatotoxic effect because cause block the respiratory function of mitochondria in the heart and liver cells. Membranolabelazing effect influence mast cell and basophils. This leads to histamine and serotonin release and as a result to allergic reaction. And endotoxin cause effect upon the immune systems. In large dose, at the peak of infection process, it suppresses the function of the immune systems. In small doses, during convalescence, it stimulates this function. It also activates the complement system into an alternative ways and stimulates interferon products which have protection activity. On this slide you see how endotoxin cause SIGN or symptoms. Upon macrophage activation, increase interleukin, tumor necrotic factors alpha, nitric oxidase, and cause fever, fever and hypertension If endotoxin are told active complement systems. In complement systems, protein C3A increase release histamine and cause hypertension and edema. If active C5A proteins of system complement increase neutrophils chemotoxis. Neutrophils is innate immune cells and increase activate tissue factors and cause coagulation cascade and can cause disseminated intravascular coagulation, DIC syndrome. And last, my student, virulence factors, bacterial biofilms and intracellular survival. Biofilms is a collection of integrating bacteria attached to the solid surface or to each other and includes in an exopolysaccharide matrix. One type of bacteria may be involved on more than one species may coagulate to form a biofilm. Bacteria in the exopolysaccharide matrix may be protected from the host immune mechanism and from the action of antimicrobial drugs. Intracellular survival. Some bacteria, example Mycobacteria tuberculosis, causative agent tuberculosis, live and grows intracellular within polymorphonuclear cells, macrophage or monocyte. Evading intracellular killing by phagocytotic cells can be accomplished by several mechanisms. They may avoid entry into the phagolysosomes and live within the cytosol of the phagocyte. They may prevent phagosome-lysosome fusion and live within the phagosome, or they may be resistant to lysosomal enzymes and survive within the phagosomes. On this, my lecture is finished. Thank you for your attention, and please don't forget to take the computer test in your personal account.